Well, thank you, Professor Ashcroft, for this nice introduction. I have to say it's an immense pleasure to be here talking about science, cutting-edge science, in this beautiful room, which it sort of brings out despite what you heard this morning, the timelessness of science. So we work on cell growth and aging control, and in particular work on the mTOR signaling pathways. And what I'd like to do today is give you an overview of our work. I won't show you any actual experiments, and it'll be a largely historical overview. And then I will hopefully get you up to date on where we are. In the first slide, I will show you what I consider an overview of the mTOR field. So this is the very first ever model of mTOR signaling published shortly after we discovered mTOR in 1991. This is a more up-to-date, but already, I would say, woefully out-of-date model. We've stopped drawing models of mTOR signaling in simple two-dimensional sheets of paper because they've just gotten too complex. On this side, I'm actually embarrassed we published this model because if you look carefully, there are more question marks here than any actual valuable information. It essentially says mTOR exists and little else. Now, this increase in complexity in this field has been driven by this exponential increase in publications in this area. So this would be, I guess, consists of an allegro phase of cell science. And this increase in publications has been driven by the fact that mTOR turned out to be both fundamentally important and medically important. In fact, much more so than we suspected when we first came across it in the early 1990s. Now, the field actually started before 1991. It started in the mid-1960s when a group of scientists went to Easter Island, also known as Rapa Nui, and what they were doing was collecting soil samples from which they hoped to isolate a bacterium which produced some novel secondary metabolite which they could then develop into a drug. And what they were looking for in particular was a novel antifungal drug, which they hoped, of course, to commercialize. They did isolate by 1975 such a molecule, which they called rapamycin, after the indigenous name for the island, Rapa Nui. And they were developing its antifungal activity for use in the clinic. But as they were doing so, they realized it had the undesirable side effect of suppressing the immune system. And needless to say, suppressing the immune system is the last thing you want to do to someone with a fungal infection. So it was discarded and not further developed as an antifungal. Now, about 10 years later, it was then rediscovered and repurposed, but now as an immunosuppressive drug. And this coincided with the advent of transplantation surgery as a viable medical procedure. Key in this, what I would call a revolution in medicine, was the ability to suppress the immune system so it would not reject the transplanted organ. And it was drugs such as rapamycin which made this possible. Now, if you go to Easter Island, there's actually a plaque which commemorates this event of picking up this handful of dirt from which they isolated the bacterium. This is shown here. It is in Portuguese, and it says, "In this place is obtained in January of '65 samples that not only allowed the isolation of rapamycin, but also led to the inaugurated a new era in transplantation therapy." So this was really a major advance in medicine. Now, having worked on rapamycin most of my adult life, I always wanted to go to Rapa Nui to pay homage at this site. And it's actually one of the last trips I did just before the COVID pandemic. We flew all the way to to Rapa Nui. We dropped off our bags at the hotel and immediately rushed out into the bush to see this site. We were very excited to be there, but when we got there, we found that the plaque had been stolen. So we never saw the plaque. If anybody's seen it, please let me know. I'm still eager to see it. But nonetheless, we were happy to be there. So what does rapamycin actually look like? So this is this molecule, small molecule rapamycin. It now has applications in 3 different major therapeutic areas. It first became very famous, as I mentioned, as an immunosuppressive drug, but now it's even more famous as an anti-cancer drug, and it's also used in the treatment of cardiovascular disease, restenosis after angioplasty in particular. So it's extremely rare that one molecule can have applications in 3 such different major therapeutic areas. And this should already be a hint that there's something, I would say, important about whatever the mechanism of this drug is, whatever the target of this molecule is, of what the target of rapamycin is. So we decided around 1990 to understand how this molecule works. It was known to have immense therapeutic potential, but the underlying molecular mechanism of how this worked was completely unknown. So we had an outstanding team working on this, a American postdoc by the name of Joe Heitman, a Swiss student by the name of Jeanette Kuntz, and a collaborator in a local pharma company then known as Sandoz, now known as Novartis, who collaborated on trying to identify the target of rapamycin. Now what they did, which was extremely unusual, was they decided to use yeast to understand the molecular mechanism of action of this drug. And this was an extremely unusual thing to do because this molecule is obviously being developed as a drug to be used in humans. And to give it to yeast cells, which is the most basic unicellular organism you can find that's eukaryotic, was viewed as somewhat as a strange idea. It was like giving aspirin to a yeast cell. Why would you ever do such an irrelevant physiological experiment? Of course, what Joe and Jeanette and Rao remembered was that rapamycin, many, many years earlier, had been first isolated as an antifungal, and yeast is a fungus. So this suggested whatever the mechanism of action of this drug, it's probably conserved all the way from yeast to human. So you could then use yeast cells to go after the mechanism, and the advantage of going after yeast cells, after it in yeast cells, is that you could then use yeast genetics. In those days, you could not do genetics with mammalian cells. You had to use more cumbersome biochemical techniques, but yeast genetics was a powerful approach in those days. And it was a very simple experiment they did. They simply poured some agar into a petri dish, shown here, plated our standard lab yeast strain on here, and what this agar had was also impregnated with rapamycin. And if there were no rapamycin in this agar, there would have been a lawn of yeast cells growing on this. But since there was that rapamycin here, the antifungal activity of the rapamycin killed all the cells we plated here, except the very few cells which were spontaneously resistant to rapamycin. So what Joe and Jeanette and Rao did was they picked these different colonies, yeast colonies, purified them, and then studied them, determined how many genes were mutated, which genes were mutated. And when we did that, we found that any one of 3 different genes were mutated to confer rapamycin resistance. Most mutations fell in this gene FPR1, and these mutations were in this gene which encoded a protein called FKBP, which I'll come to later. Rarely, we also had mutations in 2 novel genes, which we then called TOR1 and TOR2. So we had no idea why we had mutations in 3 different genes, what these genes encoded, and why they conferred resistance to rapamycin. When we cloned them and sequenced them, we understood the answers to these questions, and the answers relate to the mechanism of action of rapamycin, which is shown here. So this, again, is rapamycin, now drawn in a different configuration, a space-filling configuration. This is the product of that FPR1 gene, the FKBP protein, and what we know now is that rapamycin diffuses across the plasma membrane, doesn't have a specific transporter, which when mutated could confer resistance to rapamycin. Once inside the cell, it binds this small 12 kilodalton protein called FKBP, and this FKBP rapamycin complex then binds directly to the TOR protein. The TOR protein is extremely large. It's about 30 times bigger than this FKBP protein. So this is just a small part of the TOR protein, the so-called rapamycin binding domain in the TOR protein. So most of the FKBP is not essential for cell viability, but is absolutely essential for drug action. That's why any simple loss of function mutation in the gene encoding this protein gives complete rapamycin resistance. And this is why mutations in this gene were so common. TOR, on the other hand, is absolutely essential for cell viability. If you mutate it, introduce a loss of function mutation, the cells die and you never would have obtained a mutant. So this, the gene or this protein can tolerate very little mutation, and every mutation we obtained in the gene encoding this protein fell in one specific codon, which encoded a key contact site between rapamycin and TOR. And the effect of these mutations, of course, was to prevent the binding of FKBP rapamycin to the TOR. So these mutations turned out to be extremely informative. They elucidated the mechanism of action of rapamycin, the fact that it has to form a complex of FKBP first, and then this binds directly to TOR. Why we need FKBP, I'll come to later. We know that rapamycin can bind to TOR in the absence of FKBP, but it cannot inhibit TOR, and we will come to an explanation of that later. So the mutations elucidated the mechanism of action. They also identified the rapamycin binding site in the TOR protein. It's a very large TOR protein. But most importantly, these mutations led to the discovery of TOR itself via cloning of the genes which are now mutated to rapamycin resistance. So what did the TOR proteins turn out to be? Well, the 2 TOR proteins turned out to be 70% identical, so very highly related proteins. They also turned out to be the founding members of this class of atypical protein kinases, a kind of enzyme which modifies other proteins via phosphorylation of those proteins. Also, since this early discovery of the TORs in yeast, TOR has now been found in all organisms, all the way from yeast to human. And the name TOR, originally given for the 2 TOR proteins from yeast, has been kept for all organisms with a prefix to indicate where this protein is from. mTOR originally meant mammalian TOR, but it's now been corrupted to mean mechanistic TOR as well. Yeast turned out also to be somewhat unusual in that it had 2 TOR genes and therefore 2 TOR proteins. All other eukaryotes have a single TOR protein, and we'll come to this later as well. So this is where we were in the mid-1990s. We did know what the function of these TOR proteins was, other than be the target of rapamycin. They, of course, did not evolve to be the target of rapamycin. They had some other function in the cell. And a great deal of work from our lab showed that the role of TOR is to control cell growth. And now I have to take I have to digress a little bit to explain what I mean by cell growth, because this is one of the major steps in this research. So what you have up here is what a cell normally does. You go from 1 cell to 2 cells to 4 cells. This is how cells proliferate. And how cells divide like this was a very active area of research in the 1990s. But what people did not appreciate so much is that this process of proliferation is more than a simple division of cells, which is an increase in cell number, but it's also an increase in cell size or cell growth. And the best way to understand is if we look at these things occurring separately. So if you had division without growth, the cell would divide and get smaller, and this would be catastrophic. This would not sustain life. On the other hand, if you have growth without division, the cell would get bigger and bigger, and this would also be incompatible with sustained life. So TOR, this process is a superimposition of these two sub-processes, which I would say are equally important. Strangely enough, in the 1990s, nobody was studying this part of the aspects of cell proliferation because they thought, as you heard, growth was not actively controlled. There was no active mechanism or system to control cell growth. This was a passively regulated process which occurred when the nutrients were available. So it was controlled simply by the availability of nutrients. If nutrients were available, there were building blocks, the cells built. If not, they did not build or grow. But we discovered, after many years of trying to understand what the role of TOR might be in cell division, many very difficult years of experiments leading nowhere, we realized that by largely a process of elimination that the role of TOR is to control cell growth. And this is a major advance because it indicated that cell growth is indeed an actively regulated process. There's a mechanism there to control it. From the perspective of today, it seems very naive that we once thought something as important as cell growth was not regulated, but that was indeed the case. Now, to show you what I mean by cell growth, again, I'll show you some experiments done not by us, but by some colleagues working with Drosophila. These are much more photogenic than our experiments at the time with yeast. And in the left side here, you have, this is a tissue from a fruit fly. This is the so-called fat body, which is the equivalent of a liver for a fruit fly. And this experiment was done by Tom Neufeld, a scientist in America. And what he did was he selectively inactivated TOR protein in these two green cells. For the aficionados, these are GFP marked cells. It's a part of the experiment to know which cells, in which cells TOR had been inactivated. And as you can see, these cells end up being much smaller than the neighboring wild-type cells. Bruce Edgar did the inverse experiment. He hyperactivated TOR, and he gets the inverse result. The cells end up being much larger than the neighboring wild-type cells, again in the fat body of the fruit fly. So the obvious conclusion from this is that the role of TOR is to control cell growth and thereby cell size. And this is largely confirmation of what we had discovered earlier in yeast cells. So cell growth is actively controlled. Now, Tom Neufeld took this experiment one step further, in which he isolated a TOR mutant fly, in which all the cells of the whole animal or the whole fly were now defective for TOR signaling, or for TOR protein. And it's important to point out that this is a so-called TOR hypomorph. It's a partial loss of function. If this had been a complete loss of function, this fly never would have been born because that would have been a lethal mutation. This fly has about 20% normal levels of TOR protein. And as you can see, the fly is smaller but perfectly proportioned. And then Tom Neufeld asked the question, why is this fly smaller? Is it smaller because Is it a number issue or a size issue? In other words, is it smaller because it has fewer cells, or the normal number of cells, but each individual cell is slightly smaller? And how would you ever do an experiment to test that? Well, what he did was he looked in the wing, because the wing is a plane of cells, so you can easily count the number of cells and measure the circumference of individual cells, and from that you extrapolate to the rest of the fly. And what he found was that this fly has more or less the normal number of cells, but each individual cell is smaller to give rise to this smaller but perfectly proportioned fly. And the conclusion then is, of course, that TOR controls not only cell size, but thereby controls size of tissues and ultimately size of the whole animal. To my mind, this is an absolutely astounding result, because to make a fly like this is a program in 4 dimensions, and here this program has been changed in at least the 2 dimensions we see here by a single mutation in the genome, in this case in the promoter of the TOR gene, which reduces but does not eliminate TOR activity. You can think of all the regulatory circuits, all the metabolic circuits would have to be rewired to create this smaller but proportionally, properly proportioned fly. I never would have thought this would have been possible, but there you have it. It's also important to realize that TOR controls growth in many other physiological contexts. For example, here are some of my favorite examples. If we fast or feed, our liver changes in size, and this is due not to acquisition or loss of cells. The existing cells change in size. One example you'll all be very familiar with is muscle. If you exercise, your muscles get bigger. And again, you don't acquire more muscle cells, but your existing muscle cells get bigger. And this has all been shown to be controlled by TOR. In the central nervous system, there's some very dramatic changes of not only in cell size, but cell shape. And someone's actually even shown that queen bees are larger because the larvae which give rise to the queen bee is fed a special so-called royal jelly, highly nutritious royal jelly, which induces the TOR pathway to give this larger bee. So the conclusion from this is that TOR controls growth in many physiological contexts, and this is controls growth very widely, controls it in many different physiological contexts, and also in many different organisms. Essentially any case in eukaryotic biology where you see a change in cell size, there's a strong likelihood that TOR is somehow involved. So how does TOR actually control cell growth? Well, it does so by controlling a large number of cellular processes which collectively determine mass accumulation and thereby cell size. And these processes can be subdivided into 2 groups: the anabolic processes which TOR activates, and the catabolic processes which TOR inhibits. So what TOR does is it balances these opposing forces of synthesis and degradation such that the right amount of mass accumulates in the cell in response to, and this is important, something else we discovered in the mid-1990s, in response to nutrients. So TOR senses nutrients, activates all these metabolic pathways or inhibits metabolic pathways depending on whether it's anabolic or catabolic to lead to mass accumulation and cell growth. So the next question we addressed was, how does TOR actually control all these processes? Well, there are effector signaling pathways which emanate from TOR, and these intersect with key proteins involved in all these processes to control them. So an overview of this, a very high-level overview, can be shown here. So this is a cell. We have TOR in the cytoplasm. It responds to factors on the outside, nutrients. We also now know growth factors, and this then signals into the cell where through a biochemical pathway involving this TOR kinase, which then controls all these processes. And we can look in this now in greater resolution, and these are the experiments we then performed for about the next 10 years, again taking a genetic approach with yeast cells. And this is what we learned. This is where we were in the year 2000. we realized that the two TOR proteins control cell growth via two separate signaling pathways or biochemical pathways. One pathway contains the TOR2 protein, and this signals to the actin cytoskeleton, which determines the shape and what part of the cell grows. And we view this then as mediating spatial control of cell growth. This pathway or branch over here, which is more complex than the one over here, controls all these processes which lead to mass accumulation in response to nutrients. And we view this then as mediating temporal control of cell growth. And of course, these two aspects of cell growth have to be integrated, the temporal and the spatial, to control the growth of a cell so it grows at the right time in the right place. And what better way to do that than to have the same protein mediating both? So that's what we think is the logic behind having these two branches and these TOR proteins signaling through both. But at this point, we had many questions. For example, what determines the specificity of the two TOR proteins? Why can TOR2, by the way, we call this the TOR shared pathway because either TOR protein can redundantly function in this branch over here. We didn't know what determined the specificity. Why can TOR2 signal through both branches, but TOR1 only through this branch over here? And we also did know something which confused us at the time, is that rapamycin FKBP could inhibit TOR in this branch, but for some reason the same protein, TOR2, is not inhibited in this pathway. So what determines the specificity, the diversity, and the rapamycin specificity of rapamycin? So we decided to address these questions now by taking a biochemical approach by purifying the two TOR proteins directly out of yeast cells. And the idea was to use a gentle purification scheme, so if there are interacting proteins, we'll identify them via the co-purification of the TOR proteins, and once we have these, we can understand what TOR actually interacts with directly in the cell to mediate all these processes. When we did this, we found that indeed the TORs interact with other proteins, all shown here. TOR2 interacts with these proteins in the TOR2 unique pathway, and TOR1 or TOR2 interact with these proteins in the TOR shared pathway. And we call these complexes TOR complex 1 and TOR complex 2. So this was a major advance for us because it told us that TOR is in fact two structurally and functionally distinct kinases. And this different function and structure is determined by the nature of the two complexes, which other proteins TOR interacts with. It also explained the diversity of TOR signaling, the specificity of TOR signaling, why TOR2 can signal through both branches, because TOR2 can assemble into both complexes. TOR1 can assemble only into one of the two branches. And we also learned at this state that rapamycin inhibits only this branch because rapamycin FKBP can bind to this complex but not to this complex. And why it can bind here but not here, I will tell you shortly. Okay, so you have to keep paying attention. Okay, so this is where we were in the year 2002. At this stage, we started to wonder why or whether these two complexes were conserved all the way to mammals from yeast, like TOR itself. And at the time, we looked in there was only one genome sequence available, and it was not a complete genome. And when we looked in this partial mammalian genome, we found some proteins which had some identity with all the proteins we'd previously identified as making up these two complexes in yeast. So this then suggested the two complexes might be conserved all the way to human. We did the experiments to test that notion, and indeed we found that the two complexes are conserved all the way from yeast to human. Orthologs of the proteins we identified in yeast bind to mammalian TOR in mammalian cells, and we now call these complexes mTORC1, mTORC2 for mammalian TORC1 and TORC2. These two complexes phosphorylate or act on the same downstream proteins that the yeast equivalents interact with in yeast cells to control the same processes. So the picture that emerged at this stage is that this is a primordial or ancestral signaling network which is conserved all the way from yeast to human to control this very fundamental process of cell growth. Now, that statement I just made is not entirely accurate because this part of the network up here evolved much later. This evolved with multicellularity and was then grafted onto this part of the network down here, which already existed in unicellular yeast cells. And the reason for that is that this is the growth factor signaling pathway, for example, responds to, for example, to insulin and other growth factors. And this was this evolved with multicellularity because in multicellular organisms, it's critical to control the growth of every cell in the body with every other cell in the body. And that then is what the growth factor input provides. It provides growth control over a whole body plan. Whereas local nutrients, and now we also know local energy in the form of ATP, feed into activating the mTOR signaling network. So when these three conditions are met, growth factor, nutrients, and energy, these are all the conditions which favor cell growth. The network is activated. All the anabolic processes which it controls are either activated or inhibited and leads to cell growth. Now, another rather fascinating aspect of the mTOR signaling network is the unusually large number of diseases to which mTOR has been functionally linked. And what all these diseases have in common is they're characterized by inappropriate or ectopic cell growth, again underscoring the concept that mTOR is a master regulator of cell growth. And this can be either malignant forms of cell growth or benign forms of cell growth, but in all cases, cell growth. And more recently, mTOR has been implicated in another set of disorders, the disorders known as metabolic diseases, and those are such as obesity and diabetes. We know that chronically high circulating levels of nutrients can upregulate mTOR even in the absence of growth factor, and this can then lead to adipogenesis and obesity, which can in turn lead to type 2 diabetes. We have a more direct link to type 2 diabetes is this negative feedback where mTOR phosphorylates and inhibits this component of the insulin signaling pathway so the cell becomes insensitive to insulin. This is one of the molecular links between nutrients and insulin signaling in the development of diabetes. In this case, some have actually proposed rapamycin as an anti-diabetic drug because if you inhibit mTOR, you inhibit the negative feedback loop and you restore signaling through the insulin signaling pathway. I don't think anybody's actually developing rapamycin as an anti-diabetic because there are a number of other complications which would prevent that, but something giving credence to the notion that you might be able to do it is the world's most commonly prescribed anti-diabetic, metformin, acts at least in part by inhibiting mTOR via activation of this negative regulator. So mTOR is involved in many diseases, and what is also interesting about this image here is that all these diseases, almost all these diseases you see here, are so-called age-related diseases, which brings me to the important point that mTOR also controls aging. It controls aging of individual cells. So if here we have a young cell, active mTOR signaling can lead to the aging of this cell. And not only does it lead to aging of individual cells, it can lead to aging of entire organisms, and this has been now shown in yeast, in worms, and in flies. In 2009, there was a publication which created a large amount of excitement, was the demonstration that mTOR also controls lifespan in mammals, in this case in mice. So this is the first ever pharmacological extension of lifespan in a mammal. And this then created lots of excitement because it, number one, this finding and the fact that it can also extend lifespan in other organisms provide an explanation for what we've known since 1935, and that is that dietary restriction or caloric restriction or eating less can extend lifespan. And nobody understood the mechanism underlying why eating less extends lifespan, but now our discoveries in TOR provide the answer to this question. If we eat less, we have less TOR activity, and then TOR alters metabolism in a way according to this lower level of nutrients, and this, the net effect of this altered metabolism is, leads to a longer lifespan. So TOR controls lifespan, at least in model organisms, but the fact that it does it in mammals suggests that this could also be the case in humans. The TOR controls the aging in humans, and this, I would say, it's likely. It has not been shown. It's a difficult experiment to do to demonstrate this in humans. You cannot do a clinical trial because you can only do clinical trials with diseases to test the effect of a molecule on disease, and aging is not a disease. But the assumption is that TOR controls aging like it does in all other eukaryotes which have been examined, which means that now we have a way to manipulate the aging process, perhaps pharmacologically in humans. We can either do it by inhibiting TOR with rapamycin. This would mimic dietary restriction. And of course, you could maybe at least do it by genetic interaction of TOR complex 1. So how does TOR control aging? I told you how it controls growth, all the processes it controls to control the growth of a cell. Again, work from our and other labs have shown that the processes which TOR controls in mediating cell growth control are the same processes it controls in mediating the aging process. For example, protein synthesis, ribosome biogenesis, and these other activities. And it's a very strong correlation. The same process is controlled, TOR controls to mediate cell growth. It also controls to mediate cell aging. So that means there's a common mechanism by which TOR controls the growth process and the aging process, which means that this might explain why TOR is involved in all these age-related diseases. It's just an extension of its growth-promoting role, which when an organism is young, and then once it reaches adulthood, it continues to activate these processes inappropriately, let's say, to drive now the aging process. Okay, so in addition to understanding growth and aging at the cellular level, we also want to understand TOR at the atomic level. And this is what so we determine the structure of human TOR complex 1, and that is shown here. We learned that TOR complex 1 has 2 TOR proteins, one here and one here. It has 2 catalytic sites, one here and one here. So this is where substrates bind to get phosphorylated. We also determine the structure now of TOR complex 1 bound to FKBP rapamycin, and this then explained the puzzle of how rapamycin inhibits TOR. Why does it need to bind FKBP to inhibit TOR complex 1? And that's shown here. So this is now this FKBP protein, which TOR glues to the lip of this catalytic cleft, and as a result, it forms a lid which blocks access of substrates to the catalytic site. So TOR complex 1 can no longer phosphorylate its substrates because FKBP sterically hinders access to the catalytic site. Rapamycin alone cannot inhibit TOR complex 1 because it simply doesn't have a sufficient mass to block access of substrates to the site. We also determined the structure of TOR complex 2, and that's shown here. And what we learned from this is that the TOR protein is also a dimer of TOR and the other associated subunits which make up these two complexes. TOR itself has the same structure in both complexes, but they have different functions because of the associated subunits which are different between the two complexes. And what the structure, what we learned from this structure is what had been puzzling us since 2004 was that TOR complex 2 was insensitive to rapamycin, whether TOR complex 1 is sensitive. And the structure revealed the answer. This subunit, which is specific to TOR2, masks the rapamycin binding site, the FKBP rapamycin binding site on the lip of the cleft here, such that FKBP rapamycin cannot bind TOR in TOR complex 2, whereas it can bind TOR in TOR complex 1. So that resolved the issue of the long-standing issue of why TORC2 is rapamycin insensitive. Okay, let me just end with a photo of a collage of different members of the lab who've contributed to this work over the years. There have, of course, been many more than are shown in this slide, but of course, I thank them and everybody else who has contributed to this story. Thank you.